Welcome to Night School. I'm Darian.
And I'm Christina.
Night School is a program of the California Academy of Sciences Nightlife, a weekly in-person program for adults that mixes science, culture, and art every Thursday night, and we are the online version. Tonight, I'm really excited to meet a group of skilled biologists who work at the Academy's Steinhart Aquarium. We're going to talk about the global conservation work they're a part of. It's our aquarium without walls. In a figurative sense. I guess a literal aquarium without walls would get like wet and messy, and it's figurative speech. Anyways, we've got three phenomenal biologists who work with some of the most charismatic and unfortunately threatened species at the Academy, and we're so excited to hear about all the work they do to help their species chances of survival, whether it's in San Francisco or on the other side of the world.
Thank you, Darian. I'm glad you explained to people why an aquarium without walls would be a problem. So thank you for that. So first up, we have Lisa Larkin, who cares for the corals in our 212,000-gallon Philippine coral reef exhibit. She propagates corals in the Academy's coral spawning lab. She plants new corals, even though they aren't plants, and monitors the colony's health. And so she recently took her expertise into the field, where alongside science and conservation partners, she worked on a coral breeding project to help ensure the health of Honduran reefs. Then we have Kelsey Paulling, who was recently in Panama to bake a frog-shaped cake and celebrate Golden Frog Day. But while she was there, she worked with Valle Amphibian Conservation Center, which is a nonprofit dedicated to ensure the survival of Panamanian amphibian species, including the critically endangered golden frog. So she'll talk about how her work is supported the efforts of this small nonprofit. And then finally, we have Steven Yong, who talks about his recent work in South Africa to volunteer with SANCCOB, which stands for the South African Foundation for the Conservation of Coastal Birds. So if you've seen our African penguin exhibit, it's modeled after Boulders Beach, which is home to one of South Africa's few remaining breeding colonies. And our colony is actually part of something called a species survival plan. So Stephen went there earlier this year to specifically help with sick and injured seabirds, and our biologists often help with hand rearing abandoned African penguin chicks too. And so he'll talk about that. So great program coming up.
As always, tonight's program is live, so say hi, let us know where you're watching from, whether it's your first time joining or you're a Night School regular. We love to hear from you regardless. Tonight, we'll do something a little unusual. We'll have a group Q&A after all of the speakers go. So we'll get to hear about their experience working at the Steinhart Aquarium kind of collectively, which is cool. But still, feel free to drop questions in the chat throughout all of the presentations. We'll be sure to keep track of them and loop back around at the end. So without further ado, first up, we have Lisa Larkin.
Hi, everyone. My name is Lisa Larkin. I am a senior biologist at the Steinhart Aquarium, and today I'm here to talk about the Roatan Marine Park collaboration that we did in September of 2022. Just to give you a little bit of background on me, I am a primarily coral biologist for the Steinhart Aquarium. And one of my biggest jobs is to help care for the corals within our coral spawning lab. So the coral spawning lab is in collaboration with IBSS, which is a research division within Cal Academy. The coral spawning lab has two primary scientists and researchers. The first is the PI, Rebecca Albright, and the second is Dr. Elora Lopez Nandam, who is the postdoc for the coral spawning lab. And she is actually the person that I traveled to Roatan with, and she was my travel buddy, partner in crime, part-time Spanish translator. Yeah, it was a wonderful trip with her. Just to talk a little bit more about our coral spawning lab, we focused primarily on two species of Acropora, both from the Indo-Pacific, Acropora millipora and Acropora hyacinthus. Our main goal is researching coral genetics in aquarium spawned corals, as well as how to better rear these aquarium spawned corals and how that can translate later into off-planting corals throughout the world. So I also wanted to just toot our own horn, I guess. Elora was just published on Tuesday, and you can see the little image there on the kinship and genetic variation in aquarium spawned Acropora hyacinthus corals. So cheers to her. I don't know if everyone knows what or where Roatan is. It is a Bay Island off of Roatan, about 40 miles off the coast of mainland Honduras. It has an area of about 32 square miles, so it's that middle one there if you can see it. It has a population of approximately 110,000 people and is home as well to the Mesoamerican Barrier Reef, which is the second largest in the world. Because of this barrier reef, it's actually very common for tourism, specifically scuba diving tourism. So it's a really popular spot for scuba divers. One of the reasons Roatan was in need of coral spawning help is because of stony coral tissue loss disease. This disease was actually first discovered in Florida in 2014 and first seen in Roatan in 2020. So as you can see on the pictures on the right, stony coral tissue loss disease can affect corals really quickly. So within, from January 5th there to February 1st, you've lost 60% of the coral's tissue. The picture next to it is actually a picture I took in Roatan. So it is still an ongoing battle there. And then as you can see lower here is a picture of what was a 7-foot tall pillar coral. And pillar corals were found to be really susceptible to stony coral tissue loss disease, and unfortunately the entire colony was lost. So how is stony coral tissue loss disease transmitted? It's waterborne and direct contact. And honestly, it's not fully understood how or why it happens. The treatment of it is challenging because it's actually you have to basically paste amoxicillin onto the coral. And sometimes that treatment doesn't take it. Sometimes takes multiple treatments. So you need scuba divers just go down there and actually physically treat these corals. So it's quite challenging. The collaboration specifically was with Roatan Marine Park. It was established in 2005. It's a non-governmental organization and community-based and women-run. Woo! But it has huge operations. So they do the park rangers, which monitor overfishing. They monitor reefs for both spawning and stony coral tissue loss disease. They help with the treatment, that application of that coral paste on ailing corals. They educate the community on, why reefs are important. And they also do both coral asexual reproduction and coral sexual reproduction. And I'm about to show you what the differences of those two are. So coral asexual reproduction, this is such a beautiful photo. And I can't take credit for it, I have to say. But the first type is also known as fragmentation. So you break off a piece of a coral and you have a new coral, and you can place them on these coral trees, which you see in the picture here. And they grow bigger, and then you're able to outplant them. The one issue with this method of coral reproduction is that you lose genetic diversity. The coral that you broke it off of is a perfect genetic clone of the one that you have, the fragment that you have. So yeah, you lose genetic diversity there. So the more beneficial one would be coral sexual reproduction, which is a very complex cycle, but we're going to try and go through it relatively quickly here. So adults will spawn once a year, typically. Usually the whole reef is harmonious in this. So specifically each species is harmonious, I should say. So they release this egg sperm bundle, and it floats to the surface, is positively buoyant with water movement and motion. It disassociates from the sperm and the eggs dissociate. The species that we look at cannot self-fertilize. So we need multiple corals to be spawning on the reef to have fertilization. But once fertilization happens, we get cleavage, and you get from 1 cell to 2, from 2 to 4, and so on. Going around the circle, there's interesting phases called like the prawn chip stage, the bowl stage. Once we get kind of to this teardrop stage and the elongated planula stage is when they actually start swimming. And that swimming is for them to actually find the right place to call home, because once they settle, they don't move. They are sessile invertebrates. So they're looking for places that have good biofilm, good coral and algae and whatnot. And once they settle, they'll start splitting into multiple polyps and then eventually grow up into that big adult phase. Just to talk a little bit about the collaborators. So this collaboration was actually made specifically to help teach other Honduran organizations how to do sexual coral reproduction. It is a challenging process, and to do it without training it's challenging. So the collaborators included Roatan Marine Park, Sea Corps, obviously Cal Academy, GIZ, which is a German nonprofit organization specifically for helping Caribbean reefs, Honduran Coral Reef Fund, Marifund, MINAE, and there's more from that actually. And yeah, it was a really enthusiastic group, really good people. So we're going to start the workshop here. This first thing was actually learning how to make a coral crib. So this was done on land first, and then I'll show you pictures about when it's more together and deployed in the water. But this is going to be used for where we put that planula for them to settle onto tiles. So I'll show you that in a second. We also helped train them to make these nets here. As you can see, there's a buoy on top. So we talked about how the egg sperm bundles float. So they float to the top of this, and then there's a little tube on the top where we can collect the egg sperm bundles. We trained the folks to do this during the day because it's obviously much easier to do during the day. And then we also learned our dive site during the day because at night it gets more challenging. We then prepared the coral rearing in situ basin, or from now on I will be calling it the crib. So as you can see here on the far left, this was us with it mostly prepared. Then we had to move it on a boat to where it was going to be finally dropped into the water. And here's Alora and I in front of it once it's been deployed. We also helped with the tiles. As you can see on the left there, what we want is the corals to settle on these tiles so that we're able to easily off-plant them once they've grown big and strong. the middle section is looking into the crib, or the middle picture is looking into the crib. You can see the tiles in there and they're floating in a basket. And then on the right-hand side, you can see Elora is actually cleaning off the windows there. So this is how water exchange happens. And yeah. So in addition to doing all of this, we were also monitoring during the night. So they were long nights, but rewarding nights, I should say. Each buddy team was assigned 4 colonies to monitor both Orbicella annularis and Orbicella fabulata. We were specifically interested in Orbicella annularis, which is the upper picture there, and then Orbicella fabulata is below. So we watched the corals for 2 hours, so from 8:20 p.m. to 10:20 p.m., or once we got low on air, we would come to the surface and call it for the night. Our dive site was Bikini Bottom. It was about 26 feet max depth. The first 2 nights were no spawning. So you can imagine it was a long dive and nothing really happened. You imagine swimming in a 10 by 10 box for about 2 hours at nighttime, and you can imagine what it was like. But the following day, we had spawning. So the picture on the right shows something called setting. So when corals are about to spawn, they get the gametes just to the edge of the mouth of the polyp, and then depending on the coral, we'll release it within minutes, within a couple hours, depends. The middle picture is of the nets deployed. And then this far picture is of actual spawning. And I do have a couple videos to show you guys of spawning. And hopefully you don't have to hear me breathing underwater. So these are set polyps, and there's the spawning there. And as you can see, water motion is what moves these around. This colony we didn't collect from specifically because we were lucky enough to have a lot of different corals spawn on the reef. Let me show you one more video. Oops, let's go next. And here's another one of them spawning. Not quite as instantaneous, I would say, but still quite pretty. It is a really magical thing to watch coral spawn. And you can just kind of sense that the whole reef knows that things are happening that night. There were more like worms in the water, more mysid shrimp. There was more fish swimming around, ready for a meal. But yeah, it was really, really quite special. Oops, we're not going to try and watch that again. Okay, so we did collect from the Orbicella annularis, and then once the egg sperm bundles floated to the surface, we were able to collect them here on the upper right or upper left-hand side. You can see the eggs, sperm bundles from 5 different corals there. In the middle section of that upper row is us fertilizing. So we actually we hand mix it so that it gets that water movement, water motion, so that they disassociate. And then here is a picture of a computer, which is us looking for that cell cleavage. And then the rest of the pictures are of us purifying the eggs and making sure that they're ready to be put into the crib. So just a little recap, this is where we're at now. We're at the 4-cell stage. And yeah, they are ready to go to the crib. So there you can see us at about 2 to 3 a.m. pouring our freshly fertilized gametes into the crib. Here is Elora checking on the eggs in the crib. And then, yeah, another picture just so you can get a better idea of what we're looking at. All right, settlement. So after about a week, we carefully inspected 15 tiles from each of those little baskets. And unfortunately, we did not find settlement. With that being said, this was still a very important and this training was still so important for the other people. There could be a variety of factors why they didn't settle. I think it may have something to do with the fact that it rained quite hard while we were there. And the crib is very close to the surface, and that may have changed both the pH and the salinity within the crib and made it challenging for these babies to settle. So I also do need to say that this species has actually never been successfully settled in the crib. This species was chosen by Roatan Marine Park specifically because they knew when it would spawn. So they were able to go through the motions and hope that something good would happen at the end and train all of these wonderful people to do this on their own. But unfortunately, we did not get any settlement. So just to, talk more about Roatan Marine Park, because it is such a wonderful organization. Moving forward with them, they are looking to get permitting from the Honduran government to build an ex situ coral nursery, and then get funding for it and building the ex situ nursery, and then training staff on how to do that. So an ex situ nursery would be, just to clarify, they have a spot completely, excuse me, a spot completely picked out where they would pull water from the bay and have tanks dry on land and raise them until they're big enough, raise the babies in a lab setting until they're big enough to be off planted. So we wouldn't necessarily have to use the crib in the future. They need to train staff to care for these corals. They are looking forward already to June 2023, where they would like to spawn brain corals. They do plan to continue partnership with Sea Corps as well as hopefully Cal Academy. And they are also looking to build their network of divers so they get a better idea of when corals spawn on each reef in their area. It wasn't all work. Elora and I also had some fun dives. We were able to monitor Acropora palmata. We attended many different presentations. I even presented to a group on our work. We attended their yearly fundraiser. We even were able to go to an indigenous community there and have some delicious food. And we were also able to visit Cordelia Banks, which just has some of the most beautiful coral reef I've seen in all of the Caribbean. And with that, I thank you all for your time. And I want to acknowledge the Roatan Marine Park and all of the Steinhart Aquarium biologists, as well as Elora for being such a wonderful travel buddy. That's it.
Thank you so much, Lisa.
Yeah.
And there's a bunch of questions for you, but we will do those at the end. And so I'm going to pass, the baton to Kelsey.
Hi, everyone. Welcome. Thank you so much for listening to our fun talks about our trips outside of the aquarium. My name is Kelsey Paulling. I'm a biologist too in the Steinhart Aquarium. I work on the terrestrial team, which means I work primarily with things not in the water, so things in the rainforest, the penguins, things like that. And I had the pleasure of going to the Valle Amphibian Conservation Center in Valle, Panama, back in August of this year. I was one of two biologists. I also went with Tim Wong, who's a senior biologist on my team. And we were the first biologists from the Steinhart Aquarium to go to Panama to help at the amphibian conservation. So it was a very, very exciting trip for us. First and foremost, we went down there to work with the Panamanian golden frogs, Atelopus zeteki. These frogs are actually a toad. So all toads are frogs, but not all frogs are toads. The Panamanian gold frog is a toad that is endemic to Panama. They're only found in streams in the cloud forests. There's actually two different varieties. They have the lowland Panamanian golden frogs and then the highland Panamanian gold frogs. And we have lowland Panamanian gold frogs here at the Steinhart Aquarium. They're on the first level of the rainforest. And we are part of a species survival plan, which is why we have the golden frogs here in our care. And I'll touch a little bit more on that in a minute. But PGFs are classified as critically endangered, and they're actually presumed to be locally extinct in Panama. They haven't been seen in the wild since about 2009. And the reason for their decline is numerous, but the main ones are pollution of the waterways and also chytrid fungus, which is a fungus that I will talk more about on the next slide. So chytrid fungus, or Batrachochytrium dendrobatitis, is a waterborne fungal infection. Zoospores will travel down waterways, and when they find a suitable amphibian host, they will affect the keratinized skin cells, which in adult or metamorphed frogs is usually the underbelly or the armpits or soft tissue underneath. And in tadpoles or juveniles, it's around the mouth or feeding parts. And when these zoospores affect the skin, it causes thickening or hardening, which for amphibians can be extremely detrimental, even fatal, because many amphibians use their skin to regulate temperature, mineral balances, hydration, and some even use their skin to respire. And so during this chytrid infection, these processes are all interrupted, causing electrolyte imbalances, loss of muscle control, heart failure, and it can also present as reddened skin or sloughing off of the skin. Nearly 90 species of amphibians have led to extinction due to chytrid fungus, and it affects hundreds more. It's also found on every continent except for Antarctica, so it is a huge worldwide epidemic for amphibian species. The Panamanian golden frog SSP that we are part of is the Species Survival Plan. And there are a number of different species that we have here at the Academy that are involved in their own Species Survival Plan programs. But what a Species Survival Plan is basically a group of people that oversee all management of a particular endangered species, anything from captive breeding to initiating new husbandry protocols or increasing the success of our ways of caring for our animals, to in situ conservation and how we can support conservation from afar. So the SSP is basically a group that dedicates their time and energy into overseeing all of these different parts of animal management. So why did we choose to go to EVACC this year specifically? We have had Panamanian golden frogs in our care for a number of years and collaborate with the Project Golden Frog, which is run by a woman named Vicki Poole out of Fort Worth, Texas. And she is a well-known liaison between the Panamanian golden frogs in care in the United States and sort of collaborating with our institutions and then also in situ conservation in Panama. And we also have a croak fund here at the Academy through one of our penny crushers down in the aquarium level. So over the last several years, we've been slowly accruing enough funds to be able to send biologists to Panama and also to help support conservation efforts through our croak fund. And so this year we were finally able to put all of these things together and make a trip happen. And we chose EVACC because they are an excellent foundation in El Valle that started in about 16 years ago by Heidi and Edgardo. Edgardo is here holding a snake and Heidi's just above. They started EVACC 16 years ago and became a registered nonprofit in 2017 with the mission to become a leading organization dedicated to ensuring the survival of all Panamanian species, not just frogs, but any type of fauna in Panama that is having a hard time. And they are trying to achieve this through community outreach, education, ecotourism. Edgardo also does some private consulting for local companies that are trying to build on land. And they're doing really important work and working 7 days a week but they were very heavily affected by the pandemic, and so now seemed like a really great time to go down and help them get back up on their feet. So EVACC is in El Valle de Anton, or the Anton Valley, which is about 2.5 hours west of Panama City, up in sort of the high montane cloud forest. And these are some photos that I took of the town. It's a very vibrant place, lots of colors. And as you can see, golden frogs are very culturally significant, and they are all over the place. Even walking in the grocery store, there were frogs on the aisle placards. And so it was a really great experience to be able to see how much the people of El Valle really cherished the golden frog. El Valle was actually built in the crater of an extinct volcano. So there are very tall mountains surrounding the town on all sides. It's also a bit cooler climate, being a little bit farther away from the coast, and there's lots of rainfall. So it's a very suitable place for frogs in general, for lots of flora and fauna. And the reason that EVACC was built or originated in El Valle is because this was a location where Panamanian golden frogs were once found all over. So about 10 or 15 years ago even, if you went out and walked along a stream, according to Edgardo, you would probably see 1 or 2 dozen golden frogs bouncing around. But unfortunately today, you just don't see them anymore. So they're trying to troubleshoot how to get Panamanian golden frogs back reintroduced into the wild. So EVACC's facilities are on a hotel property. In 2019, they moved into this cabin that they call, hoping to eventually turn it into a visitor center to generate some revenue. But unfortunately, since they moved only a few months before the pandemic, and COVID restrictions were very strict in Panama, they were having a really hard time sort of getting off their feet, opening this visitor center. And as Heidi and Edgardo were really the only 2 people that were taking care and doing the captive breeding of these frogs, a lot of their time and energy went into the management of the population that they had, and didn't have much time to set up this visitor center. So that's one of the things that Tim and I focused our efforts on when we first went down there. But they have this cabin, and then behind it they have these containers that they use for breeding, for holding. There's a storage container, and also for live foods so that they can feed the animals. And then these 2 photos down at the bottom are photos of the visitor center and gift shop. And as you can notice, these tanks that they had were all completely empty when we first arrived. So it was a clean slate and lots of work to do for us. Inside these shipping containers, I mentioned one was for storage. They had some supplies that we were able to use. But these other photos are from the other 3 shipping containers. There were 2 that were dedicated to animal holding and breeding. I believe they had about a dozen different species of native amphibians that they are actively trying to breed. These smaller rainbow-colored containers are all breeding rack. So they had juveniles as small as only a few weeks old to a couple months old. And then the center photo is of the live food room. So Heidi, it's pretty much a full-time job on its own, but Heidi cultures all of the live foods on her own. She cultures both crickets and fruit flies to feed all of the frogs. And to do that is a very time-consuming and meticulous process. And that particular container was probably 100 degrees and a lot of moving parts to keep track of. So Tim and I, while we were down there, had several different tasks, but the main tasks that we had were to help build these exhibits for the visitor center. That took the first week that we were down there, most of the time. But we also got to go on some night surveys and collections to take inventory of different species and abundance in the local areas. And our trip also coincided with the Golden Frog Festival, which happens every year. This year it was on August 14th, but it's a really fun festival where the whole town turns up and celebrates the golden frog. And Heidi and Edgardo are trying to bridge the gap between celebrating a species and then also recognizing that it's a real animal that is possibly extinct and the effects that that can have going forward. So Tim and I built 9 different displays. We went through the storage container and found all of these plumbing parts and cleaning materials and everything that we could need to build exhibits from the ground up. And hopefully this will play. Oops. Oh, maybe it won't, but that's okay. But each of these 9 tanks that we built were designated for different species. So more for climbing, some were for aquatic species. And so we had to kind of make each tank suitable for whichever species was planning to inhabit that particular tank. And I should mention that Panamá, or El Valle actually, is a little bit remote and quite difficult to get supplies to. So Tim and I were able to bring some supplies with us from this photo up on the right. We brought some substrates, some electrical equipment, some filtration equipment. But El Valle, most buildings don't have addresses and they have a very unreliable mailing system. So we here at the aquarium take for granted that if we're missing something or need some supplies, we can order it on Amazon and it arrives the next day. But unfortunately, Heidi and Edgardo don't have that same luxury. And so we had to be a little bit thrifty with some of the equipment that we needed or some of the supplies that we needed. So we, Tim and I, went out on several collection trips, we called them, or plant shopping, we called them, where we went outside the building and we collected plants to use in these exhibits. We collected rocks, sand, logs, all of the above. But chytrid is still a very large issue, and we didn't want to contaminate any of the spaces that we were building. So anything that we collected from the outside had to be completely sterilized. And so this photo up at the top is a photo of us baking rocks because chytrid can't survive super high temperatures. So if we boiled things or baked them, or some of the plants we dipped in diluted bleach solution just to kill off any pathogens before we brought them into the visitor center to make sure that our frogs or any of those animals would stay healthy. In addition to building those exhibits, we also conducted some night surveys. Edgardo took us out to 3 different locations on 3 different evenings with headlamps to spotlight and see. Basically, we were trying to take inventory of how many species we could find in certain areas and abundance of those species. Like I mentioned before, Edgardo would go out in one evening and probably see a couple dozen golden frogs. He'd probably see a couple dozen different types of different species in one given night. And now we would go out in one night and maybe see 4 or 5 individuals, hopefully of different species, but not necessarily the case. But we were still able to see quite a few different types of animals and some very cool things as well. On this picture up here on the left, these are 2 different species of glass frog. And then down here, you'll see a metamorph. So that was really exciting for us to see recruitment or a juvenile. And over here on the far right corner, these are glass frog eggs. that we found as well. So we saw all 3 stages of the life cycle for that particular species, which was really exciting. We also saw some wild things like this cat-eye snake eating a lizard. And we also saw an eyelash viper, which was the first venomous snake I've seen in the wild, which was really cool. And then the last thing that we were a part of before we unfortunately had to come home was the Golden Frog Festival. So this festival happens every year. This was the first year that EVACC had national involvement, or El Valle's festival had national involvement. The ambassador of tourism came through town, which was really exciting. And that just means that the Golden Frog is getting more national recognition. But we participated in a lot of different activities throughout town. There was a 5K run. We took a cake to the preschool as they were celebrating. We also had a stand in town where some people could come and actually see live golden frogs for the very first time in person. So it was a very humbling experience to be a part of. And it's something that we hope to continue here at the California Academy of Sciences, and also in the future, celebrating Golden Frog Day and raising awareness about endangered species. So EVACC has many plans for the future. Mainly, they're hoping to develop a program for reintroduction of not only Panamanian golden frogs, but other native species to the area that are critically endangered or also locally extinct. As it stands right now, they're hoping to build small mesocosms for controlled release. But before they do that, they must first figure out a way to build immunity to chytrid fungus in the animals before they are released. And then also secure funding to be able to hire staff to then go out and monitor these mesocosms in long-term studies. So it doesn't work so well if you have a plan but then release a lot of frogs and don't have a way of measuring their success once they're reintroduced into the wild. So they want to make sure that they have that funding secured for a while to make sure that they have a successful program. They're also hoping to upgrade their facility. Right now they have dechlorinated water for their breeding containers, but not for their visitor center. And chlorine is very harmful to amphibians. So having a way of ensuring that their water is safe to use with all of their animals is really important. They also hope to expand partnerships, not only with us, but with other institutions that have Panamanian golden frogs in their care. But there are still several things for them to overcome, and for all of us to overcome, in fact. So chytrid fungus is still very prevalent on every continent, including here in California. Chytrid is here in California as well. Another thing to overcome is their access to resources in El Valle. That making sure that they have enough supplies continuously, or being able to get anything that they're low on is sometimes challenging for them. And also finding reliable and qualified staff as their facility continues to grow and their projects are expanding, ensuring that they have the staffing to keep up with those things is really important as well. So we hope to be able to continue to support them through our Croak Fund by sending biologists down in the years ahead. And I would just like to especially thank Heidi and Edgardo for hosting us at their facility, to Vicky Poole at Project Golden Frog for putting together all of the logistics of our trip and being the liaison for all of the things that are happening down there. I'd like to thank the aquarium staff here at Steinhart for covering Tim and I while we were gone, and also to Academy of Sciences members and visitors who contribute to our Croak Fund, and for listening today and hopefully passing on this information. So thank you.
Thank you so much, Kelsey. That was wonderful. We're all putting August 14th, Golden Frog Day, on our calendars.
Perfect.
Okay, thanks. We will see you in a little bit for Q&A. And right now, I'm going to bring on Steven Yong.
Hi, everyone. Thank you for joining us tonight. I have been having some technical issues, so I do apologize in advance, but hopefully this keeps on going as smoothly as it has so far. As Christina mentioned, my name is Steve Yong. I'm a senior biologist in the Steinhart Aquarium, and I'll be talking about some of the African penguin conservation work that I and another biologist did back in June, as well as kind of our penguin program. So why did we do a field conservation for African penguins? We're located in California. Actually, a lot of people who even come to the Steinhart Aquarium or the Academy of Sciences sometimes don't even realize that we actually house African penguins, and we have since 1983. They're actually not down in the aquarium. If you happen to visit us, they are up in our Tusher African Hall, which is part of our Natural History Museum. But if you're not able to make it physically to the Academy, you can check out also our penguin webcams, which you can find on our website, and those run 24/7. So when our building reopened in 2008, we also created a dedicated penguin conservation fund, which is very similar to the Croak Fund that Kelsey mentioned earlier. So thanks, Kelsey, for talking about that, as well as SSPs, so that I don't have to. So specifically what Emma Cosina, who's a biologist on our terrestrial team, and I did was that we went down to South Africa, Cape Town specifically, and worked with an organization called SANCCOB, which stands for the South African Foundation for the Conservation of Coastal Birds. I'm not sure how that acronym works out, but I'm told that it's actually Afrikaans, so it might make a little bit more sense there. But it is a nonprofit whose primary objective is to reverse the decline of seabird populations through the rescue, rehabilitation, and release of ill, injured, abandoned, and oiled seabirds, especially endangered species like the African penguin. And they do not only rehab and release those birds, but they also support many other programs to further the conservation of African penguins and other seabirds. Unfortunately, South Africa, there is a large shipping route past the Cape of Good Hope. So there are also oil spills, unfortunately. But on average, when there are no big accidents like that, SANCCOB treats up to 2,500 seabirds, of which approximately 1,500 are African penguins every year. So here's a little map of South Africa. Cape Town is found on the west coast, which is where we were, but SANCCOB also has a field station on the east side of the country in an area called Port Elizabeth, in a small town called Gabura. And then for reference, the major African penguin breeding colonies are Boulders Beach and Stony Point. Boulders Beach being the location that the Academy's penguin habitat is modeled after. So here is the lovely location of SANCCOB. That's the base of Table Mountain back there. So that is actually not the ocean. That's a saltwater pond behind SANCCOB, but they do have obviously a near access to the ocean because they are working with coastal seabirds. And here are a couple pictures of the facilities. So right now Emma and I went back in June, which happens to be, since South Africa is in the southern hemisphere, one of the slower periods of the year for their rehab. So we were fortunate enough to be there at a quieter time of year. And this is more of their facility. This is one of their pools. Obviously, being seabirds, they need to have access to water. What they also do is when they have young penguin chicks, they do need to make sure that these birds know how to swim and know how to take care of themselves. They're often given daily time, multiple times, to swim and to strengthen their swimming muscles. Because they unfortunately do have some birds that are non-releasable due to severe injuries, or unfortunately sometimes if there is too much handling, too much human involvement, birds can become imprinted and lose their fear of humans, which makes it an issue for their own safety if they are released. So they have something that's called a home pen, which is a residential area for a rather large colony of penguins, as well as some other birds. For instance, on the right there, you can also see a gull. You can also, if you look closely, see a couple of rockhopper penguins, which are actually not native to South Africa, but are often carried in by cargo ships. Here are some of their facilities that we worked in. Food prep is obviously very important because they need to feed the large number of animals every day. The main diet of African penguins in particular in South Africa are sardines and anchovies. So they go through a lot of fish. Fortunately, they do get a lot of donations of fish as well as money. And some of their other more important areas of the facility are the chick rearing unit, because chicks have often not, much like human babies, do not develop a strong immune system until after a few months, up to a year of life. So they are reared in a completely separate building by separate staff. And then, like I mentioned before, unfortunately, there are occasionally oil spills, and oil is rather toxic. Unrefined oil is rather toxic to animals and humans. So there are these specific wash bays where they can clean up those soiled birds. So myself and Emma Casina went down in June. So which is actually the middle of winter, like I mentioned. So we spent a lot of time with various species that SANCCOB works with, for instance, the rockhopper, gulls, and it was obviously very cold and rainy as well. And here, over the course of the year, these are the different species that are the majority of the intakes that SANCCOB does. So, as I mentioned, the kelp gulls. There's also gannets. There are multiple species of African penguins, depending on the age of the birds as well as where they're from. PE stands for Port Elizabeth, which is that eastern colony. CT is Cape Town, was where we were. But there's also crowned cormorants, which are also an endangered species, as well as Cape cormorants, which are endangered. So Cape gannets are an endangered species. They're actually fairly large birds. You can see they're actually they're diving birds. So they don't just dive from the surface of the water. They will actually dive from flight into the water to catch their fish. So they will enter the water at speeds of up to 75 miles per hour. And in order to be able to do that and not inhale water, they actually don't have external nostrils, which is something very interesting about them. They usually lay their eggs in September, October, but unfortunately, due to availability of food, sometimes they've had adults lay much later in the year. And then those late breeders, because they are unable to find food before the chicks are able to take care of themselves, have to abandon them. These are some of the birds that we saw. See on the left, Em and I started calling them Muppets because of their fuzzy, kind of awkward gangliness. And then they grow into these dark, sleek birds as they are juveniles. And then adults, they are this very striking white bird with very, very pale blue eyes. And some more of those Muppets that we were working with during our time there. And here is a video of Emma. Oh. Let's see if we can get that video to play by any chance. Oh no. I guess I'm also having video issues. Oh. Well, moving on. Some of the other species that we work with are kelp gulls. Kelp gulls are not endangered. They're actually fairly common, much like gulls are with any coastal city, but SANCCOB does work with them anytime there's injury. And unfortunately, kelp gulls, one of their favorite foods is the trash that humans produce. And that trash, unfortunately, has a lot of issues and causes botulism, which if you ever had food gone bad and tasted it, that's the bacteria that makes you sick. And so a lot of these kelp gulls will eat this tainted food, and then it causes nerve damage because it is a neurotoxin. And so these birds, we have to actually give them charcoal that absorbs that toxin, and lots of fluids, and it takes probably about 2 weeks for them to recover. Fortunately, there's about a 75% release rate. The cormorants, during our time there was only one of each species. The one on the left is the bank cormorant, and the one on the right is the Cape cormorant. You can see they both have foot issues. Often things like that are caused by what's called ghost fishing gear or ghost nets. So netting that has been abandoned by fishermen that is left in the ocean, or is floating around, or it gets washed onto the beach, and animals get tangled up in it. We also had Prince Charles while we were there, the great white pelican, who was brought into the facility because he had a broken wing. He was his namesake because the staff at SANCCOB felt that they shared the same hairstyle, which I can see the resemblance. And you can see how very large Prince Charles was. He was a good 3.5 feet tall, but surprisingly only weighed about 18 pounds. So those flighted birds definitely have much lighter bodies and bones than penguins do. And of course, African penguins. During our time there were about 60 juvenile penguins that had been raised from eggs in the on-site chick rearing unit. And then since we were there in June, which is the early part of winter in South Africa, there were some early winter storms that washed out several penguin nests due to the availability of food. A lot of penguins are laying late eggs, and so normally animals don't reproduce in the fall or winter due to the availability of food. And so this is an issue for one of the reasons why penguins, they're getting a lot of abandoned penguin chicks. So this is a lot of our day. We are feeding juvenile penguins. We're doing a lot of cleaning. And again, it was quite rainy on Sunday, so we were utilizing the same heat lamps that the baby penguins were using to stay warm. And you can see here's an adult on the right side, again with an injured foot. I believe this particular bird was actually attacked by a dog. The African penguin colonies are actually on the beach, very near a lot of residential areas, and where people live, they often have pets, and unfortunately, outdoor dogs and cats often find penguins to be easy prey because they do not move very well on land, as they are much better built to be swimmers. And unfortunately, this is another video that won't play, so we'll skip that. We also got the opportunity to visit both of the breeding colonies. Stony Point is the place that we were actually allowed. We went to release some rehab birds, and we obviously had to go to see Boulders Beach to see the inspiration for our very own habitat. The Stony Point penguin colony is the only colony in South Africa that's actually been increasing in population for African penguins, and it's 10% of the global African penguin population. The scientists estimate that since the 80s, the population of African penguins has dropped from 100,000 breeding pairs to only about 20,000 breeding pairs, and they could be completely extinct by the year 2035. So the work that SANCCOB is doing is very important if we want to continue to see African penguins in the world. So this is us boxing up the boxes as well as releasing them. And then this chart also shows all the number of chicks admitted and the ones that were released. So you can see that SANCCOB has a fairly good success rate with releasing penguins. Each penguin that is released is also microchipped. So if it ever happens to return to SANCCOB, they are able to track them. And here are some pictures of that Stony Point. If any of our viewers are from California, it actually might look very, very familiar. The climate of South Africa is actually very similar to central California. You can see penguins. We always talk about penguins living in cold climates, but you can see here it is a very green, sandy environment. And here you can see why it's called Stony Point. Some very interesting rock formations. And you can see actually kelp beds, much like the ones you find off the coast of Monterey here. And then here, yep, no snow or ice on for these penguins. They're living on a nice green beach, but you can also see they are nesting here. Nesting on sand makes it really difficult to maintain a nest when it rains. So this is how, unfortunately, their nests get washed away. Their normal habitat is building nests or burrows out of guano or their own waste, which actually hardens up really into a really hard substance. Unfortunately for them, penguin waste, guano, as it's called, is actually very high in nitrates, which makes it a very, very good fertilizer. So a lot of African penguin habitat has been destroyed by humans who are harvesting or mining the penguin waste for fertilizer. Some more lovely pictures of Stony Point. And unfortunately, another video that won't play. And then myself and Emma at Boulders Beach Penguin Colony. The gentleman in the picture with us, his name is actually Vard. Not only was he a fellow volunteer at SANCCOB, but his day job is also as a penguin ranger, which I'll talk about in a bit. You can see Boulders Beach, again, very similar to Stony Point in that there is a large sandy area that a lot of the penguins are nesting. Fortunately, even though this is a very well-known tourist spot for folks to visit penguins, humans are only allowed to walk on this boardwalk, so it prevents people from harassing the penguins, but you can see that they are still rather close. And here you can see some juvenile penguins. They're still in that fluffy stage, which means that they actually haven't developed their waterproof feathers, which means that they are still relying on their parents to feed them. And again, this was in June, which was the start of winter. So chicks of that age normally should be found in the late summer, which would have been probably more February or March. So they're a few months late. You can see in this picture, those white items in the back are actually some artificial nest boxes that SANCCOB has put out, and that the Academy has actually funded research to design and build those artificial nest boxes because we prefer the penguin not nest on loose sand, which allows their nest to be washed away. And the penguins do prefer a nice little burrow. And so we have worked with SANCCOB and some other organizations around the world to develop some nest boxes that have been fabricated out of fiberglass to be more weather resistant, but also provide the right habitat for a pair of penguins who might be raising chicks. Some more pictures of cute penguins, as well as a hyrax. If anyone is familiar with the fauna in Monterey, you might notice marmots in Monterey, and this is the South African version of a marmot called a hyrax. And here is the inspiration for our own habitat of Boulders Beach with these very, very large boulders. And then some more pictures of the lovely penguins on their sandy beach. You can see the kelp forests, which again are very, very similar to the ones that you might find in Monterey. And this would have been another video. So beyond what we do to send staff down to SANCCOB to help physically rehab birds, we want to continue to do that, and that's part of the Penguin Conservation Fund. What we also do is we want to provide healthy and safe breeding grounds for penguins, which are those nest boxes. There are, I believe, currently a couple hundred that have been deployed between Stony Point as well as Boulders Beach, but they obviously need more. We also want to provide to rehabilitate the natural habitat. So a lot of the natural habitat of African penguins has been destroyed due to erosion because of construction for human property, for heavy winter rains, as well as stormwater damage, again from human construction, as well as trampling, and a lot of invasive plants. And then how I mentioned before, the ranger project, we do, the areas that the penguins do live in are protected by rangers, but those rangers are responsible for the entire area, not just the penguins. So they are responsible for enforcing rules, doing property and habitat restoration for a much larger area. So the goal for SANCCOB is to train up a team of rangers who are specifically there to work with penguins as well as the other seabirds. So those rangers are being trained to be able to evaluate issues with penguins and be able to call SANCCOB in for intake, as well as they are specifically trained to rehabilitate the natural habitat specifically for penguins. And that is something that the Academy is very involved in, as one of our goals is to regenerate that natural world. I do want to thank you, all of our viewers, as well as the Academy for supporting myself and Emma, as well as two other biologists who are just finished up another session with SANCCOB, Sparks and Spencer, and then as well as Lisa, who happens to be my coverage partner. So while she was working in Honduras and I helped cover her work here, she did her part by helping to cover my work while I was in South Africa in June. And I thanks all of you, as well as our members, for contributing to our mission, as well as our Penguin Conservation Fund.
Thank you so much, Stephen, for that presentation. I think we're going to have all of our biologists back for our Q&A segment, which I'm very much looking forward to. Our audience has asked some great questions. I think first and foremost, the audience, and then I would also like to pair it. Like, thank you so much for this amazing conservation work you do. We all just thank you. It's great to hear about. So first, I'm going to ask a question. I'm going to pose it to everyone. What came first, your working with the specific animals that you work with, or your love for them? Did you specialize as a biologist in the animals you took care of first, or it's kind of a chicken or the egg question?
I can answer that first. My career has sort of taken different veer-offs as certain opportunities come up. I personally studied reptiles and amphibians in college, and that's something that I've always tried to veer my career towards. But then when I got a job here at the Academy, we have such a diverse collection of animals that now I work with penguins, and I have a jellyfish in the venoms gallery, and a lot of invertebrates. And so I think probably most of us can relate that different types of animals come up and you accept those challenges because it's fun to learn new things. But we all have sort of those animals that we started off with that we are really passionate about and like to hold on to as well. So for me personally, amphibians and reptiles are that passion species, but I love having sort of the experience with a little bit of everything else.
Great. Does anyone else have anything to add?
No, I think she said it well. A lot of us have a variety of different displays, exhibits, habitats, whatever you'd like to call it. But I think for me, I've always been very driven with corals, I would say. And yeah, that's mostly what's in my exhibits. I don't have quite the variety that Kelsey has.
Great. Well, Lisa,
I feel like I'm kind of.
Sorry, please go, Steve.
Actually, a little both, because my background is actually similar to Lisa's, where I work mostly with fishing, corals, and invertebrates. But when it came to the Academy, I wanted to challenge myself and work with animals that I'd never worked with before, and that was the first thing that was. And that was either Claude or the penguin. I'd start small with penguins first. And now I've discovered I enjoy working with those little birds, or those little well-dressed footballs.
Yeah, I could only imagine how much fun that must be. Lisa, I have a question for our audience, has a question for you, actually. Lydia asks, how do different corals know how to spawn all at the same time?
That's a great question. And it's actually something that we tried to mimic in the coral spawning lab. So basically, I think the biggest driving factor would be the moon. So we know that a certain number of days after the full moon during a certain time of year, specific species will go off. So I can even speak for, currently our lab is in the middle of spawn watch for corals that were originally found in Australia. So We know at least 5 days after the full moon that they'll spawn, but again, there is a window. So from 4 days ago till 4 days from now, we'll be checking the corals for spawning.
Oh, wow. It's exciting.
It is exciting. I'm like nervous. They got to spawn.
It might happen.
Anytime now.
Let's see. Kelsey, why has chytrid become such an issue for amphibians, particularly recently? Is it invasive? Is it a new threat?
Yeah, that's a great question. It was only discovered in the late 90s, I believe. And because it's a waterborne pathogen, it is very easy to transmit. So any waterway that's affected, it can track on shoes. So even people traveling from one continent to another, if you're wearing muddy boots or anything like that, it can transmit in that way. But unfortunately, there was a lag between when it was discovered and when people first noticed amphibians being affected by it. And it's pretty much amphibians exclusively because of the type of amphibious skin that they have, the really porous, really, just very delicate type of skin that they have, that when it embeds, it can throw off their entire body function. So there was a lag. People started noticing that frogs were turning up dead, or that frogs were looking very red and irritated and really struggling. Before, that was a couple of years, I think, before they discovered that there was this actual fungus that was causing it. And by that time, chytrid had already been transmitted to several different places in the world. So it is one of the leading research topics for any type of amphibian conservation, is kind of pinpointing how chytrid, kind of how it started, how it's transmitted, and how we can sort of get one step ahead of it and create immunity before more amphibian species are targeted. Because some amphibian species are really susceptible, some aren't, and we're still trying to figure out exactly what it is that makes certain species really susceptible.
Mm-hmm. Yeah. Once an individual is infected with chytrid, is there any cure? Is there anything you could do on like an individual level to help?
Yeah, there are some antifungal treatments. The name of it is escaping me right now, but there is an antifungal treatment that has been shown to help recover from an infection. But it's the same, as some species are more receptive to that treatment than others. And also trying to figure out a way to sort of get that immunity, almost like a COVID vaccine or a COVID booster before something is exposed to chytrid, and kind of creating that baseline immunity.
Oh, great. I believe one of our audience members specifically asked about vaccinating against chytrid, so you have your answer. Steven, let's see, what precautions when you're taking care of penguin chicks do you need to take to make sure they don't get imprinted on you?
SANCCOB does it a little bit. It's a little bit more extreme because they are releasing their chicks back, their animals back into the wild, and so if there is a reminder that they're working with these animals, and cute and cuddly as they are not pets. So we should not be treating them like that. So they have very strict diets. We have very strict handling protocols. We try not to speak very much around them so that they don't get rated to human voices and associate humans with food, because they are not going to have people hand feeding them as much as some people would love to do that. So yeah, it's trying to make sure that they associate humans with food, and making sure that they stay wary of humans.
Oh, I hadn't even considered that even just hearing a human voice could impact that.
Yeah.
Let's see. A question again for all of you. Do you have like a standout or favorite memory from being in the field? Any like stories that you can tell from your expeditions?
I think mine would have to be the actual spawning events. They're just like out of this world crazy, like Pandora kind of crazy. So it's such a wonderful experience. And I don't think many people even know that corals spawn like that. So it is just such a rare and cool event. And yeah, special.
Yeah, those videos were magical.
I would have to say that I think it was the last night that we went out night surveying. Edgardo was pointing out certain areas where he'd seen things in the past, and we were traveling through an area that looked kind of mossy, and he had been talking about how eyelash vipers tend to like mossy areas because they blend in a little bit better. And I turned right when he was saying that, I turned my head and I was like, there's one probably, I don't know, 2 arm lengths away. And I probably should have been more scared than I was seeing an eyelash viper that close to my face. But it was kind of this magical moment of like, I don't know, just finding something in its natural habitat that you have never seen before. And it was close to midnight and maybe a little bit groggy, but super excited to be out there.
Steven, do you have any stories?
I do. It's the place where the animal live in the wild, and as biologists, when we are building our habitats at the aquarium, we try to make them as natural and as similar to their habitat, their wild habitats as possible. And being there in person and seeing kind of what inspires us to take care of these animals is really what really does it for me.
Yeah, I bet. Okay, we'll do another round of individual questions. Let's see, Lisa, how do you plant corals? Is that similar at all to like grafting plants? I know they're animals. Are there any similarities?
You know, Christina made that joke at the beginning and then I didn't explain it, and my bad. But so basically you saw those tiles, and those tiles, the settler will grow on either side. And what we'll do is we'll actually use like an underwater epoxy and glue them specifically on the reef using information like PAR, you know, where other corals of that species are found. And they're able to, this epoxy cures underwater. And so you just glue it right there and that's where it lives. Yeah.
Wow. Neat.
Planting.
It's like your own little garden. It's a reef.
Yeah, exactly.
Kelsey, in the beginning you said that the 2 major threats to lots of amphibians are chytrid and pollution. Are there specific water pollutants that are most dangerous or most pressing?
Yeah, fertilizers are a huge one. So any runoff from agriculture into waterways, that's a main one. Also runoff off of roads, so like gasoline, anything that's emitted from your car, oil that will wash into waterways as well. Those are some of the more major ones in more heavily populated areas or in places closer proximity to humans. But also, yeah, just I think runoff is probably the main one. And then also, you know, some people dump trash close to waterways, and trash, plastics, microplastics, things like that, breaking down can also really harm amphibian species.
Mm-hmm. And just because I'm curious, what makes a toad opposed to other frogs?
Great question. Yeah, a lot of it is morphology. So frogs tend to have drier skin. They tend to be a little bit more terrestrial, whereas frogs have much smoother, slimier skin and tend to be a little bit more aquatic. But it's also the evolutionary lineage that toads are this cluster that are encompassed by all frogs. Similar to how all newts are salamanders, but not all salamanders are newts. There's a little bit difference in the texture of their skin, in the areas that they're found, and just general behavior and morphology.
I am learning lots about amphibian clades and groupings today. Thank you for that. Stephen, let's see. Tom wants to know if avian influenza has been a problem in South Africa with the birds you take care of.
It has. Back in June when Em and I were there, it was just starting. Fortunately, avian influenza does not affect penguins as hard as it does other species. Unfortunately, birds, the flighted birds such as gulls and the cormorants, are definitely more susceptible to it. So either of those, we're taking great care to test for the avian influenza before they were even allowed into the facility because it's so transmissible between birds. Sparks from Steinhart has just gotten worse. They have found avian influenza to be present in the Boulders Beach African penguin colony, as well as I think Robben Island is another penguin colony. So far, I don't think Stony Point has been affected. And I believe the last number I heard about a month ago is that they've lost somewhere between a dozen and 2 dozen penguins to avian influenza. But right now, the SANCCOB is running a completely separate off-site facility, specifically treating birds with AI, so that's to introduce it into their main facility, because obviously they are rehabs that are already immune compromised, so that an introduction of AI would be very, very bad for them.
Mm-hmm. Yeah. And James asks, do you have any data of how many released penguins that SANCCOB releases survive in the wild?
They are working on that data, because unless they get animals that return to be treated, there's not a lot of data. Right now, one of the things also that is part of the Ranger project is to set up RFID readers, so radio frequency ID tags, those little microchips that are implanted in all released birds, so that whenever a bird with a chip swims within a certain distance from the reader, it will take that data. Unfortunately, that equipment is very expensive. So I think there's only maybe 1 or 2 that they've installed. But if folks want to actually go to the SANCCOB website, they produce their annual report. They're very transparent about their successes, as well as what their money does. And actually, if anyone happens to make it down to Cape Town, they will also take on international volunteers. So if you want to help rehab birds and handle fish, you can sign up to volunteer at SANCCOB.
Wow. Well, if I ever make it down to South Africa, Cape Town, I know what I'm doing. Okay, so our last question, I'll pose it to the entire group again. Is there anything that you've learned while out in the field that impacts the way we care for our animals at the Academy, or vice versa? Is there anything we've learned while taking care of our animals at the Academy that impacts our conservation in the field?
Kelsey, or I
Yeah,
just keep going first. I don't want to keep
sure. I
going
mean,
first.
so many things. I think we really strive here to make our exhibits as true to a natural location or a natural habitat for our particular animals. And so for me, being in Panama and seeing the natural habitat of the Panamanian golden frogs and all of the other animals that are around there with them, kind of made me realize just the like small niche that they fill and all of the things that they require to survive and all the things that we need to replicate for them here in our care. And also having all of the other biologists in our department, all of their knowledge and learning from them and learning about the way that we care for our animals here made it so much easier for Tim and I when we were building these exhibits for EVACC's Visitor Center to put all of those things in play. And I feel like they went really well hand in hand, that we had some prior knowledge and experience from our animals here to help them get everything built and started in Panama, and then also learning how they care for their animals and treat their animals and have everything in their natural habitat to bring that here so that we can replicate it and kind of showcase it to our visitors. And this is what their natural habitat looks like, and this is where you might find them. It would be kind of silly to put a Panamanian golden frog on dry sand with like a cactus, but you know, having fresh flowing water and all these tropical bromeliads and things like that, that's really what their natural habitat looks like.
Yeah, I think I would have to add, like, very similarly to what Kelsey says, we use very similar techniques, and it's obviously different when it's in the wild and not an aquarium with walls. But yeah, we use similar techniques, and it's honestly always so incredible to be able to collaborate with these people and to be able to have discussions about how they do things versus how we do things, and what's the pros and what's the cons of both of our ways. So it will help in the long run to kind of share that kind of information, for sure.
I just have to add too that we are very conscious of biosecurity and cross-contamination, and just how easy it is to pass pathogens from one exhibit to another or one species to another here in our care. And we're very careful about using gloves and washing our hands and using tools only for one exhibit, and we have a completely different set of tools for a different exhibit, and then taking that sort of mindset of cross-contamination and biosecurity to Panama, where chytrid is super prevalent. And if you walk into one of those containers with mud on your shoes, something I didn't mention in my slides is that during the pandemic, Heidi and Edgardo had 2 mass mortality events of all of the amphibians in their containers. They lost close to half of their population. And that's something as simple as not changing your shoes or not washing your hands when you come from outside to going inside. And that's something that's, I think, a skill that really helped us going down there, kind of understanding how fragile it is and how careful you have to be with biosecurity.
Yeah, important lessons to learn, definitely.
Mm-hmm.
Steven?
I think for me, it's also very appreciative to that the Academy feels that just educating our visitors and the people around the world about these animals, though important, that us making a difference in the actual conservation out in the wild for these animals is so important, because like we mentioned before, it's sustainability at a certain point isn't going to be enough, and we just need to start making a conscious effort to improve the situation, and that's what we're trying to do. And I feel fortunate to be able to bring that experience back and kind of talk to our visitors and the people watching, bring them more of a firsthand experience and understanding about the animals that we work with.
Yeah, I bet. And I know for a fact that our audience has appreciated you sharing your expertise here as well. So thank you so much, all of you, for your presentations and just for sharing your knowledge and the work you do overall. I'm going to bring Christina back and we'll, oh, there you are.
Thank you so much to Kelsey and Lisa and Steve. Yeah, say hi to them if you happen to visit and you see them walking around. But our biologists are so busy, so just extra thanks to them for like taking the time to talk about their work, because I think a lot of us who work at the academy, like we see the amazing work they do on the floor and with our animals, but we don't always get insight into like all this incredible other work that's going on. So so we here at Night School are also very happy and have learned a lot. So anyway, thank you for being with us here tonight. Our next program is on December 1st, and we're calling it we call it Night School, World's Most Endangered. So I have a question for Darian. Darian, do you know what the most endangered group of organisms is on the planet?
Is it cicadas?
That's so close. You had an extra syllable. It's cycads.
Oh.
So yes, cycads, a little bit cuter than cicadas, I think. So cycads are plants. They look like palm trees, but they have like kind of more squat and bigger, stouter trunks. And they have really spectacularly large cones. So, and they've also been around since before the dinosaurs. So we're gonna, the next Night School is gonna be dedicated to those plants. They're wild. Yeah. And then we're closing out the year on December 15th with something a little different, a very special Night School that is close to Darian's heart. And it's gonna focus on the intersection of art and the environment, and specifically about how artists and writers use their medium to process their feelings and hopes and fears about our planet's future. And so we're really excited for this, something a little different to close out the year.
Yeah, I'm excited too. As always, subscribe to the California Academy of Sciences on YouTube, so you'll be notified that whenever we're in session, you'll be notified. You'll get a little alert. Recordings of every Night School stay on our YouTube channel without walls, which means that they never go away. So you can feel free to rewatch old episodes at any time from anywhere on Earth.
Yeah, no paywalls, never.
No pay. It's all here. It's all free. It's so.
Awesome. Well, thanks again, everyone. Thank you to our regulars. I see you, Tom and April. Thank you for tuning in week after week. And to all our new viewers, have a good night and a great weekend.
No.